G.N. Ramchandran, an outstanding figure in the field of protein structure, was the founder of the Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of proteins through the use of Ramchandran plot rank amongst the most outstanding contributions in the structural biology. While at Cambridge, Ramchandran met Linus Pauling and was deeply influenced by his publications on model of alpha helix and beta sheet structures that directed his attention to solving the structure of the collagen. Cell, the unit of life. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a cell, complete structure of cell, does not ensure independent living. Hence, cell is the fundamental structure and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement, leading to the electron microscope, revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodor Schwann, 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animal and plants are composed of cells and the products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as to how the new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from the pre-existing cells, omnis cellula cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory the final step. Cell theory as understood today is: first, all living organisms are composed of cells and products of cells. Second, all cells arise from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called the nucleus. The nucleus contains the chromosome, which in turn contains the genetic material DNA. Cells that have a membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cells in the living state. Besides the nucleus, the eukaryotic cell have other membrane-bound distinct structures called organelles, like the endoplasmic reticulum, ER, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both prokaryotic as well as eukaryotic. Within the cell, the ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplasts in plants and mitochondria and on rough ER. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasma, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell in the egg of an ostrich. Among multicellular organisms, human RBCs are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cell. They may vary greatly in shape and size. The four basic shapes of the bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. Genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome/circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformations with foreign DNA. Nuclear membrane is found in eukaryotes. No organelle like the ones in the eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have some things unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosomes is the characteristic of prokaryotes. They are essentially enfoldings of the cell membranes. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram, that is, those that take up the Gram stain are Gram positive and those that do not are Gram negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be loose sheath called the slime layer in some, while in others it may be thick and tough called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in the cell wall formation, DNA replication, and distribution to the daughter cell. They also help in respiration secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contains pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from the cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three-part filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made up of special proteins. The fimbriae are small bristles-like fibers sprouting out of the cell, and in some bacteria they are known to help attach to the bacteria to rocks in steams and also to the host tissues. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S unit, which when present together form 70S prokaryotic ribosome. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA to form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. They are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophycin granules, and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. The genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plants and animals are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cell. On the other hand, animals have centrioles which are absent in almost all plant cells. Let us now look at the individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies of cell membrane, especially in human RBCs, enabled the scientists to deduce the possible structure of the plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with a polar head towards the outer side and a hydrophobic tail toward the inner part. This ensures that the nonpolar tail of the saturated hydrocarbon is protected from the aqueous environment. In addition to the phospholipid, the membrane also contains cholesterol. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte or RBC has approximately 52% protein and 40% lipids. Depending on the ease of extraction, the membrane protein can be classified as integral or peripheral. Peripheral proteins lie on the surface of the membrane while the integral protein are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as the fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of the functions like cell growth, formation of intercellular junctions, secretions, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on the either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called as passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower. Water may also move across this membrane from higher concentration to the lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against the concentration gradient, that is from lower to the higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport, example Na plus K plus pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from the mechanical damage and infection, but it also helps in cell-to-cell interaction and provides barrier to undesirable molecules. Algae have cell walls made up of cellulose, galactans, mannans, and mineral like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is the layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connects the cytoplasm of the neighboring cell. Endomembrane system. While each of the membranous organelles is distinct in terms of the structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system includes endoplasmic reticulum (ER), Golgi complex, lysosome, and vacuole. Since the function of the mitochondria, chloroplast, and peroxisome are not coordinated with the above components, these are not considered as part of the endomembrane system. The endoplasmic reticulum. Electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Hence, ER divides the intracellular spaces into two distinct compartments, that is luminal inside ER and extraluminal cytoplasm compartments. The ER often shows ribosome attached to their outer surface. The endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous in the outer membrane of the nucleus. The smooth endoplasmic reticulum is a major site for lipid synthesis. The animal cells lipid-like steroidal hormones are synthesized in the SCR. Golgi apparatus. Camillo Golgi first observed 1898 densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase and the concave trans or the maturing phase. The cis and trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cells. Material to be packaged in the form of vesicles form the ER fuse with the cis phase of the Golgi apparatus and moves toward the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Lysosomes. These are membrane non-vesicular structures formed by the processes of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Vacuoles. The vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other material not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuole can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitate the transport of the number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not visible easily under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter of 0.2 to 1.0 micrometer, average is 0.5 micrometer, and length is 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with dense homogeneous substance called the matrix. The outer compartment forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfolding called the cristae, singular crista, towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called the powerhouses of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosome 70S, and the components required for the synthesis of proteins. The mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be easily classified into chloroplast, chromoplast, and leucoplast. The chloroplast containing chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, the fat-soluble carotenoid pigment like carotene, xanthophyll, and others are present. This gives the part of a plant a yellow, orange, or red color. The leucoplasts are the colorless plastid of various shape and size with stored nutrients. Amyloplast stores carbohydrates, that is starch, example potato. Elioplast stores oils and fats, whereas the aleuroplast stores proteins. Majority of the chloroplasts of a green plant are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell in the Chlamydomonas to green algae in a Chlamydomonas, which is the green algae, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membrane sacs called thylakoid are present in the stroma. Thylakoids are arranged in stack like the pile of coin called grana or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids enclosed a space called the lumen. The stroma of the chloroplast contain enzyme required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecule and ribosome. Chlorophyll pigments are present in the thylakoids. The ribosome of the chloroplast are smaller 70S than the cytoplasmic ribosome, that is 80S. Ribosomes. Ribosomes are the granular structure first observed under the electron microscope as dense particles by George Palade 1953. They are composed of ribonucleic acid RNA and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunit. The two subunits of 80S ribosome are 60 and 40S while that of 70S are 50S and 30S. The S, Swedberg's unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosome are composed of two subunits. Cytoskeleton. An elaborate network of filamentous proteinaceous structure consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluids. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscope study of the cilium or a flagellum shows that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubule and a pair of centrally located microtubule. Such an arrangement of axonemal microtubule is referred to as 9+2 array. The central tubule are connected by the bridge and is also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus there are 9 radial spokes. The peripheral doublet are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. Centrosomes and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other in which each has an organization like a cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with the tubules of the peripheral triplets by the radial spoke made up of protein. The centrioles form the basal body of cilia or flagella, the spindle fibers that give rise to spindle apparatus during cell division in animal cell. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Singular is nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear phase space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules take place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect name of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, example erythrocyte in the mammal and sieve tube cell of vascular plant. Would you consider these cells as living? The nuclear matrix or a nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger or more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 46, that is 23 pairs of chromosomes. You will study the details of DNA packaging in form of chromosome in class 12th. Every chromosome visible only in dividing cell essentially has a primary constriction or the centromere on the sides of which disc-shaped structure called kinetochores are present. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. Some metacentric chromosome has centromere slightly away from the middle of the chromosome, resulting in one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming extremely short and one very long arm, whereas telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells.